Hello, I'm Ho-Jun Lee, and today's topic is quality control of NF1 omics data, which were used in my thesis. In our quality control pipeline for short-read DNA sequencing, we implemented a three-step QC process. Starting with the sequence reads, we performed quality assessment of the raw data. This was followed by variant calling, where we examined the reliability of our variant detection. Finally, we focused on validating possible NF1 causing variants. While we have already covered the variant calling statistics in our previous discussion, I'll now focus on the quality metrics of our sequence reads and validation of our key pathogenic variant. Looking at our base quality and GC content distributions across read positions, we observed significant fluctuations in both metrics within the first 14 bases. Additionally, we noticed a sharp quality drop in the last base. Based on these observations, we implemented trimming of these problematic regions, removing the first 14 bases and the last base from each read. We also filtered out reads with mean quality scores below 20, which affected approximately 100 reads per sample. After this trimming and filtering process, we checked that graphs for base quality and GC content became flatter than before. After trimming and filtering, we compare the sequencing metrics before and after processing. As expected, the mean number of bases decreased from 118 gigabases to 106 gigabases. Interestingly, we observed slight decreases in both mean quality score from 34.2 to 34.1 and the percentage of bases of over Q30 quality score from 90.3% to 90%. This minor reduction can be attributed to the trimmed regions actually having higher quality scores than some of the retained bases. However, given the significant GC content fluctuations in these regions in the previous slide, the trimming was necessary for the maintaining data integrity. Finally, we performed detailed quality assessment of our candidate NF1 pathogenic variant. In our patient sample, we detected both reference A and alternative G alleles at the variant position, confirming the presence of the variant. The depth of 26 reads and genotype quality of 99 indicate high confidence in our variant call. While the earlier valence of 0.26 might appear slightly low compared to the theoretical heterozygous ratio of 0.5, it exceeds the minimum threshold of 0.2 to 0.5 used in several pathogenic variant studies. Importantly, examination of parental and proband samples showed only reference alleles with sufficient depth and quality, validating this as a de novo variant. Moving on to our long-read DNA quality control, I need to make a correction to my previous statement. In the earlier video, I mentioned that our long-read DNA analysis started from pre-processing circular reads to generate hi-fi reads. However, this was incorrect. We actually received hi-fi reads directly from our sequencing provider. Following our QC pipeline shown here, we started with quality assessment of the hi-fi reads. While we also planned to evaluate SV or structure variance calling results and possible NF1 causing SV in this video, the SV count analysis was covered in our previous video, and as mentioned before, no NF1 related structure variants were identified. Therefore, I will focus on the quality metrics of our hi-fi reads. For our hi-fi read quality assessment, we compared our matrix with Novogene's example data as they were our sequencing provider. From two flow cells, we obtained 43.5 gigabases of hi-fi reads, representing 14.5x coverage of the human genome. This output actually exceeded Novogene's example dataset of 38.4 gigabases. Looking at other quality metrics, our N50 read length of 20,308 bases was comparable to their example of 20,345 bases. Similarly, while our mean base quality of 32.5 was slightly lower than their 34.1, it falls well within the expected range for PacBio hi-fi reads. These metrics collectively indicate that our received hi-fi reads were in high quality and suitable for downstream analysis. Finally, let me present our long-read RNA sequencing quality control process. Our pipeline consists of three main stages: initial sequenced circular reads, hi-fi read generation, and ultimately aligned unique transcripts. For quality assessment, we focused on two key aspects: the quality metrics of sequenced reads and the number of identified unique transcripts. For long-read RNA analysis, we initially aimed to use PacBio CLR or continuous long-reads technology with a target of 20 gigabases per sample. However, during the initial CLR sequencing, we encountered an unexpected challenge that one of our samples yielded only 8 gigabases of data, despite having sufficient RNA input that passed our quality controls. In response to this situation, our sequencing provider suggested switching to another method. We accepted the alternative method to the PacBio CCS method, or circular consensus sequencing. This technology works by generating multiple passes of the same molecule to create consensus sequences. The sequencing run generated 513 gigabases of circular reads, which were processed to yield 5.4 gigabases and 4.7 gigabases of hi-fi reads in samples with a mean quality of Q41. After aligning our hi-fi reads to the reference genome and the GENCODE transcript database, we used a collapse strategy to identify unique transcripts. As illustrated in the figure, transcripts with distinct regions remain separate, while identical transcripts are collapsed into a single entry. This process yielded 128,697 unique transcripts from our proben and 110,599 from our patient sample. To validate these numbers, we compared our results with a PacBio demo study that identified 162,290 transcripts from a single flow cell. Since we used one flow cell for both samples combined, we performed a joint analysis that identified 181,464 unique transcripts, slightly exceeding the demo study's numbers. This aligns well with PacBio's official documentation, which suggests that typical unique transcript counts range from 100,000 to 200,000 per flow cell. We used these transcripts to investigate the transcriptomic effects of our key pathogenic variant. In conclusion to this presentation, our comprehensive quality control across short-read DNA, long-read DNA, and long-read RNA sequencing showed that all our data met the expected quality standards with no significant anomalies that would affect our downstream analysis. Thank you for your attention.